This is collaborative work with some folks at Merck. We're going to present it at JSM this year, and the publication is in prep. The objective is to optimize an early development plan. It focuses on phase 1B to 2B, since the phase 3 timelines are relatively fixed. The question was, how can we leverage the relationship between early biomarkers, and now we're talking about the fourth kind of biomarkers, that is the biomarker that gives you an indication that the drug is working. So it's like an invalidated surrogate marker. Surrogate carries with it a lot of rigorous validation information, but you may have an idea that a surrogate might work. It might be like the target enzyme in the pathway of the drug. So how can that relationship be leveraged to optimize the phase 1-2 development program? How can different designs in phase 1B for proof of concept and dose finding improve the design of the phase 2B trial? And how can we use and optimize that development program to make the probability of picking the right dose for phase 3 maximal? That's what this is about. Only a couple more slides. This is the framework. We assume 2 biomarkers and the clinical response at an early time point. And we assume the correlation structure among those. The biomarkers are highly correlated. The 2 biomarkers are correlated to a degree of 0.8, and they're mildly correlated with the clinical outcome. And then we assumed underlying Emax models for each of the biomarkers and the clinical response. The first trial is a proof of concept trial. It's high dose versus placebo at 20 patients on high dose, 10 on placebo. And then after that trial is successful, we're going to add doses, each at 20 subjects, and then another 10 with placebo. And then we apply go, no go rules, and we combine the data for those first 2 trials of the biomarkers, fit the dose responses, and use the relationship to pick the doses in the phase 2 trial, and also whether or not we go to that phase 2 trial. And then we run the third trial, which is the 2B trial on the clinical endpoint, and we either do 3 or 4 doses plus placebo. And now we're going to simulate whether we need 25, 50, or 100 patients. And then after that trial finishes, we pick the dose for phase 3. So we have this simulation structure, go, no go rules set up, and now we can play plug and play rules about sample size, whether we do the phase 2, the trial 1 at a short term or a long term. So the structure looks like this. We have the biomarker POC trial, we get the results, we compute some posterior probabilities of response for the 2 biomarkers. If it's high, we go to the phase 2B trial directly. And then we use the data and simulate the phase 2 trial, get the results, pick the dose for phase 3, but we also apply a go, no go rule before phase 3. That's a rapid to phase 2 option. We also have this slower to phase 2 option where we do a dose finding with the biomarker. And we do that if we don't have such high posterior probability of response in the biomarkers. And then we do dose finding, and then we go to the phase 2B trial. And obviously, if the posterior probability is too low, we stop. All of this is set up in a trivariate normal Bayesian model framework. And the details will be in the publication. It's too much to go through now. We did simulation cases where we looked at a 2-day biomarker study versus a 4-day biomarker study. The 4-day biomarker study let us add an early observation on the clinical endpoint because 2 days was not enough. And then we looked at yes or no, do we need this dose finding trial with the biomarkers? And we looked at whether we used the posterior probabilities to determine which path we went through to the dose-finding biomarker trial or straight on. And we looked at forcing through the biomarker dose-finding trial always. The key decision metrics were the rate of simulations in which we made the correct go, no-go decisions and the probability of choosing the correct dose. I'm going to summarize the findings here. We found out that the 2-day biomarker trial is sufficient for making go, no-go decisions. You didn't need the 4-day trial. So that was important to the client because they didn't have to spend the additional money on the 4-day trial. How well did the biomarker dose finding help at improving the probability of getting the correct dose in phase 2B? Turns out that the relationship between the biomarkers and the clinical endpoint was too weak to be useful. So the client was able to skip that biomarker dose finding trial. And they had rationale for that. The project team argued we need the biomarker dose-finding trial. And the modeling and simulation group with whom we were working, we set up the simulations, and they showed no improvement over picking the correct dose with that trial. So we're actually able to save an entire trial and eliminate it from the development program. So we had a shorter proof of concept trial and straight to 2B. And those were the simulation results. The rest of the slides are the tables summarizing those results. I'm not going to go through those because we don't have time, and the slides are on the web. You could look at them, and you could listen to me talk again if you want as well. I talked about what the impact was. Biomarkers are useful for for go, no go, but when they're correlated to this weak degree, they didn't inform on dose finding. There's one finding that's really important, and whoops, I'm going the wrong way. And that is, just in general, we simulated with decreasing variance as a surrogate for increasing sample size, because the increased sample size, the runtime was too long. And we showed that much larger sample sizes than just detecting difference between drug and placebo is required to do effective dose selection. That was another important finding. We have a long list of potential work that I'm not going to go through. That could be extensions of this work, but that's just a list right now, and we're waiting for application of it. So, to sum up, biomarkers in this circumstance were useful for POC, but not for dose finding. And the fastest path was direct to 2B after proof of concept. And if you wanted to repurpose that dose finding trial, you could use it to study other regimens or determine if higher doses gave substantially greater efficacy, but you could do that in tandem with the phase 2 trial.